হ্যালো মাই ডিয়ার ভিউয়ার্স, জুলজির সেমিস্টার 3 এর বায়োকেমিস্ট্রি পেপারের ফার্স্ট ইউনিট থেকে আজ আমরা উইক ইন্টারঅ্যাকশনস ইন aqua systems ডিসকাস করব। বেসিক্যালি ইন aqua systems weak interactions are fundamental to maintain the structure, stability and functionality of biological molecules. These interactions collectively enable biomolecular folding, assembly and interactions essential for life processes. The major types of weak interactions include hydrogen bonds. Hydrogen bonds are basically dipole-dipole interactions involving a hydrogen atom covalently bonded to an electronegative atom, that is either oxygen or nitrogen, and another electronegative atom. They play vital roles in biomolecules by stabilizing the secondary uh, alpha helices or beta sheets and tertiary structure of proteins through interactions between carbonyl oxygen and amide hydrogen or amino acid side chains. In nucleic acids, the hydrogen bonds between complementary base pairs A, T and GC ensure the stability of DNA's double helix. Additionally, they contribute to enzyme substrate binding, antigen antibody recognition and specificity. In water, hydrogen bonds are responsible for its high boiling point, cohesive properties and solvent capabilities for polar and charged molecules. Ionic interactions are basically electrostatic forces between oppositely charged ions or charged groups on molecules, stronger than hydrogen bonds, but they are weaker than covalent bonds. Their strength depends on the dielectric constant of the surrounding medium, which is high in water, reducing their effectiveness. In biomolecules, ionic interactions stabilize the tertiary and quaternary structure of proteins through bond between positively charged residues such as lysine or arginine and negatively charged residues such as aspartate and glutamate. These ionic interactions or ionic bonds also facilitate protein DNA binding via interactions between negatively charged DNA phosphate groups and positively charged amino acid residues and play a crucial role in regulating ion flow ion channels. In solution, dissolved ions like sodium and chloride ion interact with water, influencing osmotic balance and biochemical reactions. Van der Waals forces, they are basically weak non-specific interactions arising from temporary dipoles caused by fluctuating electron distributions in molecules. They include London dispersion forces, which result from transient dipole in nonpolar molecules, and dipole-dipole interactions between polar molecules with permanent dipoles. In biomolecules, these forces stabilize densely packed regions in proteins and macromolecules and also play a crucial role in ligand receptor binding by enabling complementary surface interactions. Despite acting over short distances, their additive nature significantly contribute to the stability of macromolecular assemblies such as protein-protein interfaces. Hydrophobic interactions occur when nonpolar molecules or groups cluster in aqueous solution to minimize the disruption of hydrogen-bonded water networks. This process is driven by the energetically unfavorable ordering of water molecules around nonpolar molecules, leading to their aggregation, which reduces the exposed surface area, increases entropy, and stabilizes the system. In biomolecules or in aqueous system, hydrophobic interactions play a crucial role in protein folding by sequestering nonpolar side chains inside the protein and exposing polar ones to water. They are also essential for the formation of biological membranes where the hydrophobic tails of phospholipids aggregate to form bilayers, and for stabilizing lipid-protein interactions and the self-assembly of micelles and vesicles. In conclusion, the weak interactions in aqueous systems, ionic interactions, hydrogen bond, van der Waals forces, and hydrophobic interactions underscore the subtle yet profound forces that govern the behavior of biological molecules. While individually weak, these interactions collectively enable the complex folding of proteins. They the formation of stable membranes, and the precise highlights the unique properties of this universal medium in fostering life. By orchestrating dynamic yet stable molecular assemblies, these weak forces form the foundation of biological structure and function, unveiling the intricate wonders of water mediated systems. Thank you. All the best, my dear students.